Hello guys, my name is Dr. Varun and this is day 8 of 30 days, 30 most important topics for biology NEET 2025.
Chalo, let's complete it.
So guys, in this video, we're gonna be talking about certain cell organelles from the chapter Cell, the Unit of Life. So in this chapters, what are the important organelles? Three different organelles topic we're gonna be studying in it because they are little tough to understand, or maybe there's a one question which came in the syllabus which was beyond NCERT at that point, but it's not beyond NCERT. Let I will tell you. So the organelles that we're gonna start with is the ribosome. Now everyone will say that, sir, we know everything about ribosomes. But really, do you know everything about ribosome? What you don't know? Okay, in ribosome, we know that the ribosomes are made up of two things. One is rRNA and the proteins. Okay, but then did you remember that in your 12th class, there is a topic where the RNA polymerase 1, 2 and 3, when they are making rRNA, they are making different types of it. Like they are making 5S rRNA. The S is the Sedgwick's unit, which means their unit of measurement. Along with that, they are making 5.8S RNA. They are making 20S rRNA, 23S rRNA. What are these things? How they are present in the part of our ribosome? That we have to do it and understand very importantly, because question will definitely higher chances of coming in the paper. So let's just see. Till now we know that the ribosome has two parts. Okay, what are the two parts of the ribosome? One is small unit and another is the large unit. And they join together and form a single unit. Okay, so they join together and forming what? A single unit like this. So smaller unit and the larger unit. Now, in both prokaryotes and eukaryotes, different types of this joined together unit weight goes. One is 70S rRNA and another is 80S rRNA. So the first thing we have to remember that in prokaryotes and eukaryotes, ribosomes are different. So in prokaryotes, together we have 70 Sedgwick's unit ribosome. Whereas in eukaryotes, we have 80S or Sedgwick's ribosome. Now let's just come to the important point. It's smaller unit and the larger unit. Let's first talk about the smaller unit. The smaller unit, always remember, the smaller and the larger unit, they will be made up of two things. One is rRNA and the proteins. How many proteins present? This also we have to remember. So we will write it that there is rRNA that is present here. Here also there will be rRNA present over here. Then there will be number of proteins present here. Here also certain proteins will be present. Similarly would be for the larger unit. When it comes to the larger unit, there also there is rRNA present. There is protein present. Here also rRNA present and a protein present. Now just remember that what will be present there. Now if we first understand about the protein, then in the smaller unit, that is what its value in case of prokaryotes is what? The smaller unit value is 30S. Whereas the smaller unit value in eukaryote is what? 40S. Whereas larger unit in the prokaryotes, the unit is what? 50S. Whereas eukaryotes, it is 60S. One thing we will remember. The next thing that we have to remember is the rRNA which is present. Proteins. So first protein would be what? Here 21 proteins present. Here 33 proteins present. Here we have 34 proteins present in the larger unit of the prokaryotes. Whereas here we have 50 proteins present. Okay, one thing is over. Let's see other. Now how many rRNA or which type of rRNA is present? So in case of smaller unit, only one rRNA present, and that is what? That is 16 Sedworks unit rRNA present. Whereas in eukaryotes, it is 18S or Sedworks rRNA present. We will remember it. I will give you a trick after it also. If you write like this is making it understanding better. Now in the larger unit, there are 2 rRNA present. So I will write it what? That is, there is 2 rRNA types present over there. Let's just see what are they. So the first one is the 5 Sedworks unit rRNA present, and another one is the 23 Sedworks unit rRNA present. Whereas in the eukaryotes, we have total 3 types of rRNA present. Let's see that what are they. The number one, there is what? Similar like 5S rRNA present. Then we have 5.8S rRNA present, and we have 28S rRNA present. Now let's just see the trick of it. If you write like this, it's very easy. So first you have to remember what? Till now, if you have done NCERT, you know that the prokaryotes has what? 70S. The eukaryotes have 80S. They have what? Smaller unit also present, bigger unit also, larger unit also. The smaller unit of the prokaryote is what? It is 30S. Whereas smaller unit of eukaryote is what? 40S. Then we have larger unit of prokaryotes is 50S, and the smaller unit is what? 60S. Then the only thing you have to remember first would be what? That number of proteins in all of them are different. So 21, 33, 34, 50. 21, 33, 34 and 50. Then comes the rRNA. In the smaller unit, you remember in the eukaryotes, everything ends with 8. Okay, how we will remember it? See the eukaryotes. In the eukaryotes, the smaller unit rRNA is 18S, means 8 coming. So eukaryote is 8 because 88. Whereas this would be lesser than that. So if I'm saying lesser than that, it means 16S, little less will be there. Of course, prokaryotes will be smaller. They are primitive. Okay. Moving on, in the larger unit, if you see that there is what? Both have 5S, means same. Then we know that what sir told, Varun sir told that 8 will come in the eukaryotes. So here would be what? 5.8. Here would be what? 28. Okay, how I will remember 5.8 will be there? Because 5, which is less, was present there only in prokaryotes. So in eukaryotes, there will be more than 5. So 5.8, it is more than 5. 8 is also present. Whereas when it comes to 23 and 28, we know that 28 is coming means it will be eukaryotes. Whereas 23 would be what? Prokaryotes. This way, if you remember it, you will remember everything about it. So this is very important question they can ask you. They can give you that out of which of the rRNA is present in prokaryotes. You know that 8 is not present in prokaryotes. Okay, so we will write these ones. Okay, that's about the ribosomes, which is very important. Next thing we gonna understand about centrosome, centromere, centriole, and how they make the cilia and flagella. Okay, so the next organelle we gonna be, or terminologies that we're gonna be studying, what we have: centromere, we have centrosome, and we have centriole. Okay, common thing, mistakes can occur. So quickly we will see it. First of all, what is centromere? Centromere means what? Like in a chromosome, this is a chromosome. This is what? This is the primary constriction. Means the chromosome has been constricted over this area. So this area which has primary constriction present, that area is known as what? Centromere. Around centromere, proteinaceous covering kinetochores are present. Centromeres are those. What are centrosomes? Whenever the word zome is coming, means remember it that it is something related to organelle. Okay, so centrosome is the organelle. Now this centrosome is made up of centriole. Okay, so it is what made this. So normally what is centriole? Centriole is a cylindrical organelle which is present only and only in the animal cells. No plant cell have this. So if the plant cells do not have centriole, means they do not have centrosome also. But centromere will be present in every cell because chromosome constrictions, primary constrictions are present there. So this is an important point to remember. Moving on, this centriole, I said that it is cylindrical. So it must be made up of some basic units. So what is the basic unit? The basic unit is the microtubules. And these microtubules, further basic unit is what? The basic unit is the tubulin protein. Tubulin protein. Tubulin protein. So this thing you have to remember. So that is given in NCERT that the microtubules basic unit would be what? It is tubulin protein. Tubulin proteins forms a structure called as microtubules. Now these microtubules arrangement is very important. So let's just make you see that how this thing will look. So there is what at the center there is a structure, proteinaceous structure present called as hub. So this white color thing which I have made is what it is hub. I am making centriole. Okay, so this is your hub. Now around hub there is a 9 evenly spread. Evenly spread means equidistant spokes are present. So we call them what? Radial spoke. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, like this I have made it. Now these spokes are attached to the peripheral tubulin proteins which we have talked. So periphery has what? Tubulin proteins present. So let's just see that. So they are attached 1, 2, 3, 4, 5, 6, 7, 8, 9. Now this peripheral tubulin protein, the yellow color I have made, they are present in the form of triplets, means they are 3. So 1, 2, 3, 1, 2, 3. 1 is already there, 2, 3. This is 2, 3, 2, 3, 2, 3, 2, 3, 2, 3, 2, 3. Okay. Now these proteins are connected with each other also. So let's just connect it. So they are connected like this. So this kind of structure is formed. So this is how a centriole look like. Now when the one centriole is the mother centriole. During the time of cell division, it multiply into another one. So means a mother centriole will form what? A daughter centriole. So when the two centrioles are present together, they are called as centrosomes. Okay. So what we have? This is, if I am making, this is the one centriole. Another centriole is present over here. This centriole is connected with this structure. So means mother is connected by this. So I will say that what? Mother is connected with umbilical cord, the daughter. Okay, so these are what? These are the connections, fibers which are connecting mother, one centriole to the another one. They are present at the 90 degree to each other. Outside in this area, a amorphous fluid is present. Means imagine that certain viscous fluid is present over there. We are for imagination. Okay, certain things like jelly kind of thing around it, protective present. This thing is called as what? Pericentriolar material. That's it. Okay, and this when you will see, you will observe this during the time of cell division. Now more thing is that what this centriole do? This centriole help in the formation of what? This, okay, I'm making it over here, that this centriole has 3 functions. It will make cilia, it will make flagella, it will make spindle fibers. What are spindle fibers? These are the ropes which will be gone and it will catch the kinetochores. What were the kinetochores? Kinetochores are the proteinaceous flap present around centromeres. Okay, that's why the terms are joined together. Okay, so spindle fibers will be there which will observe during the time of cell division. And which time of cell divisions? During the metaphase, okay, or the late prophase. Okay, they will be appearing and they will be joining. Means appeared there first, but they joined at that time. Okay, so now the cilia and flagella, if they are being made by centriole, means their structure would have something like this, right? So that structure you have to remember, very easy to remember, to be very honest, and you can link it with. So in the like outside periphery, if there are what, if they are made by this structure, and it has what I told you, that these are what, 9, 1 2 3 4 5 6 7 8 9. So means outside certain 9 microtubules will be there. Okay, so here what will happen, outside on the periphery I will make 9 such. Now the major question is, are they also in the triplet form? No, they are doublet form. Okay, so 1 2 3 4 5 6 7 8 9. Okay, so these are okay, I will make it better. Wait. So we have what, 9 of them. 1 2 3 4 5 6 7 8, and this thing we have to just remove a little, 9. Okay, now they are not in the triplet form, they are in doublet form. So doublet form, let's make it like this. So I made it doublet. Next question would be, this not the only thing, in the center also they are present. Okay, but center they are in the single form. Okay, they are not doublet over here. Now they are joined together. This is joined by the bridge. Okay, these are joined with each other, which is called as linker protein. Okay, like this, linker protein. Meanwhile, these things are joined with, like over here a sheath is present. Sheath means covering. So a central sheath is present, this thing, the white color one. And all these outside area will join in the center also, like this earlier one, radial spoke. Here also these radial spokes present. So the arrangement is like this. Now the major question is, they will ask you like, what kind of microtubule arrangement seen in centriole and what kind seen in the cilia and flagella. In cilia and flagella, we see what, outside periphery has 9 doublet plus 2 microtubules present in the center. So this arrangement is there. Whereas in terms of centriole, there are 9 present triplet, but in the center nothing is present. No such microtubules are there. So this 9 and 2 representing what? Microtubules. And microtubules are made up of what? They are made up of tubulin protein. That's the only thing you have to remember. Rest, that they have a plasma, the cilia and flagella has plasma membrane, center part is called as exonema. Exonema have this structure, is basic thing which you can understand from the NCERT. Well, that's all for today's video. I hope that these organelles you are able to understand. If there is any other organelles which you feel tough to understand, maybe about the cis transphase or Golgi bodies, I will make an extra video for you. Just comment me down. So that's all for this today's video. Most important organelles, confusing organelles has been told to you or explained to you. I hope that you must have understood it. If you understand it, give it a like. Subscribe to this channel for more such videos. We're gonna be doing on 15th of April a very important questions topic from 11th class, whole 11th class. 3 most important topic in each chapter. Don't miss out that video also. So subscribe it, keep the notification bells on, and if you find this video good enough, then share it with your friends. Thank you. We meet soon.